I wanted to make sure that the mRNA is safe in humans. Dr. Benjamin Weider from the dermatology department has injected me mRNA coding for firefly luciferase. So this mRNA is translated into a protein that emits light, like the firefly does.
Back in 2003, Steve Pascolo made history as the first person ever injected with messenger RNA.
But it wasn't until the COVID-19 pandemic that mRNA really burst into the spotlight, offering up a potential treatment beyond infectious diseases.
There is a theoretical solution for any disease using mRNA. Any disease you can think about, autoimmune, autoinflammation, degenerative, injuries, even aging.
In this episode, we'll explore why it took so long for mRNA to be taken seriously. We'll hear Steve's story as its first human test subject, and we'll look ahead to the exciting possibilities this technology could unlock.
I'm Kai Nussbaumer.
And I'm Micah Schweitzer. This is Balancing the Future from METTLER TOLEDO.
On this show, we delve into the world of science and technology and explore its transformative impact on our lives.
Just for context, Steve, what is mRNA and how does it work in the body?
So mRNA in the body is a copy of a gene that is contained in DNA. So any cell of our body contains all the information to make anything, an hepatocyte, a neuron, a skin cell of the skin.
That's Dr. Steve Pascolo. He's a senior scientist in the Department of Dermatology at the University of Zurich, and the former co-founder of CureVac, a biopharmaceutical company pioneering mRNA research.
CureVac is where Steve was working when he was first injected with mRNA.
But of course, each cell type, depending on what it has to do, will use some of the genes, not all of them. And this decision is made on mRNA. So for example, a cell of the skin will copy the gene coding for keratin, so that the skin cells make keratin, but other cells will not make keratin. So mRNA is in every cell. It makes the cell being specific, expressing the genes that it needs to express, and it's transient. So the mRNA is always made, is translated into proteins, and then it's degraded. So you have a dynamic whereby the cells can also then adapt. If you have a wound on the skin, then the cells will start to make mRNA coding for proteins that will repair the skin, for example, and this is transient.
So before COVID-19, the discussion around mRNA was relatively obscure. So what took so long for this technology to gain widespread recognition?
The whole problem of mRNA at that time was the prejudice that people said it's fragile. As I said, mRNA is transient. In the cells, it's made and then it's degraded. So we have a lot of proteins in the body called RNAs. And RNases are proteins that degrade mRNA. And when you work in laboratory with mRNA, you have to put gloves, you have to have special solutions, special plastic, which are free of RNases, because RNases are everywhere, on every surface, in your body, everywhere. And so people knew that it's difficult to work with mRNA. So when you say, I will make a drug with mRNA, most people say it's impossible, it's fragile, it's terrible to work with mRNA. In the lab, you have to have all specific things. How would you want to make a drug with something that is so difficult to work with? And this is a misunderstanding of people doing research or medicine. They don't know most of the time how you produce a drug. And when you produce a drug, you have gloves. When you produce a drug, you use special plastic. When you produce a drug, you use special solutions. It's, of course, in the lab, it makes the life a bit harder to make mRNA than DNA or proteins. But in pharmaceutical production, whatever you produce pharmaceutically, you have seen people are very protected. The whole space is very clean. So the production of mRNA as a drug is not more difficult than producing a peptide or DNA. In a lab, yes. But people making decisions are people eventually coming from the lab or from the clinics.
My understanding is that you're the first person who ever received an mRNA injection.
Yeah, that's correct. So CureVac, we had 3 goals. So one was to optimize the mRNA vaccine because it was working, but not as good as, for example, protein vaccines. So we wanted to optimize it. The second goal was to produce it. At that time, we went to companies doing pharmaceutical products. We asked them, could you do mRNA? And then all of them, they said, you guys are nice, but you're completely crazy. There will be no market ever for this mRNA molecule. So we improved the mRNA vaccine, the production, the formulation. We did the pharmaceutical production in CureVac. So we made clean rooms and I designed a suite. It was like 6 years of work. But we finally got the certification for mRNA production in pharmaceutical grade. And then going to human was to make clinical studies in cancer patients, because this was initially my goal, to induce T-cell responses in cancer patients. And so we had everything ready for the clinical studies, but I wanted to make sure that the mRNA is safe in human. It was safe in animals, and also I wanted to make sure that it's functional. It was really a clinical study in the dermatology clinic of the University Hospital of Tübingen, where Dr. Benjamin Weide from the dermatology department has injected me mRNA coding for firefly luciferase. So this mRNA is translated into a protein that emits light, like the firefly does. So I think I'm the first human also who has expressed firefly luciferase. So the idea was
and did you light up, or did?
Yes, yes. At least pieces from my skin. So we injected in my skin, and then a few hours later or 1 day later, we repeated a few times. We looked at the site of injection. I didn't have side effects. I didn't feel fever or something like that. So of course, we recorded the side effects. And then at the site of injections, or away from the site of injections, we made punch biopsies. So you take small pieces of the skin under local anesthesia. And then in the biopsies, we could record in a so-called luminometer. It's a machine that measures the light emission. Then we could record light emission from the skin taken at the site of injection, but not away. We made small holes more and more away from the site of injection, so we could show that the expression of this firefly luciferase was at the site of injection of the mRNA and not away. We could monitor that it was coming quite quickly and disappearing after 2 days. We published that in 2007. So these are published data that I sent it to big journals. They said, "You are crazy guys. What are you doing?" So
even though you lit up.
Yes.
And you would think that even just that would get some attention, right?
We were young. We didn't have political network, to publish high. You need to have like a big name, or you need to know the editors, or to invite them, or whatever, and I was very young. So I sent it, to big journals, and they immediately said, "Crazy you, go away." So then I went down with the impact factors and finally published it in a lower impact factor. But I think if I would have been somebody a little bit famous, or it also helps if you are in a big American university, or, in a site connected to some journals, you see some people manage to publish their things.
In the time between Steve's injection and the COVID-19 pandemic, researchers worked to make mRNA stronger and safer, tweaking its chemical building blocks to reduce inflammation, cleaning up impurities, and developing lipid nanoparticles, or LNPs, to deliver it efficiently into cells.
These LNPs would later prove critical in making the COVID-19 vaccines possible. By the 2010s, animal studies and early human trials showed that mRNA vaccines could protect against viruses like flu and rabies.
In 2019, scientists had all the tools ready for rapid vaccine development: safe, optimized mRNA, effective delivery using LNPs, and evidence that it could trigger a protective immune response.
So when the COVID-19 pandemic hit, mRNA vaccines could be developed quickly and effectively. So fast forward to 2021 or 2020, what changed? Why did the pandemic have such a dramatic effect on the uptake and recognition of mRNA vaccines?
I need to mention that in 2001, we were partner of the EU grant for vaccines against porcine flu. At that time, there was a fear that there would be this kind of flu coming from animals. And so there were some programs to develop vaccines. We joined a program where they tested peptide, virus, protein vaccines, and I could add mRNA vaccines in it. So because we already postulated in 2000 that mRNA could be good in case of pandemics. So we had that in mind in January 2020. Then people thought, okay, let's do mRNA coding for the spike. And they just, within few days, basically got it. And while the others doing proteins or peptides took months to eventually design, produce, and test their protein. So the speed of production, what we have claimed already 20 years ago, was there. And it was funny to see in, let's say, February 2020, nobody knew about mRNA except the small group of people doing mRNA vaccines. And then in October 2020, everybody was speaking about mRNA. It has gone from night to spotlights within 4 or 5 months. This is rare for technology. Normally technologies, monoclonal antibodies, siRNA, always take quite some years and they move through the studies and the public starts to get acquainted, the medical people start to get acquainted with the technology. And here, within 6 months, a technology that nobody knew, including medical people, suddenly they were all exposed to it, and then exposed first in terms of information and then in terms of being really injected with it.
So after scaling up production and millions of people being vaccinated with an mRNA vaccine, what were the learnings from that? Whether good, bad, were there any necessary changes or adjustments that needed to be made?
So you can always improve vaccines. We spoke about the injections I received in 2003. We were happy there were no side effects, but we were also a little bit concerned because we were like, if you activate your immune system, you should feel a bit tired, you should feel a bit fever. And I felt nothing. And the first clinical study in patients was also, safety was super. But when you do vaccines and there is no systemic side effect, you think your vaccine might be a bit weak because a strong vaccine will, at least in some people, if you inject 10 or 20 people, you should have some people reporting some kind of side effects, local or systemic. The side effects of the COVID vaccines compared to other vaccines are very good. So the immune response is very strong. One thing that can be improved is the duration of the immune response. So we have seen that the immune response induced by the mRNA vaccines, as they are so far, is vanishing away within a few months, is decreasing. And this is the same with many vaccines. People forget, but the flu vaccines, the protein-based flu vaccines, also give an immune response that vanishes over time. So it's not something new that some immune response you get by a vaccine is tending to go down. And mRNA platform is so versatile that you have many ways to make mRNA vaccine. And there is one way called self-amplifying mRNA vaccine, and this is done by several companies, including one called Arcturus in the US. And this kind of replicating mRNA vaccine has shown to give more prolonged immune response. So I think for prophylactic vaccines, in the future, we may switch from the non-replicating, so mRNA that gives a protein and goes away, to a replicating mRNA, which is a kind of mini virus in a way. And this works really well. You need less. The injection is only a few micrograms, like 5 micrograms. The normal vaccines with mRNA are like 30 micrograms. And what Arcturus has shown, its vaccine is approved. And they have shown that the antibody response remain high for a longer time using this replicating mRNA compared to the previously approved mRNA vaccines from BioNTech, Pfizer, and Moderna. So I think, there are still improvements you can make to keep the safety, to keep the efficacy, but to increase maybe the duration of the immune response. And people need to know that mRNA is a platform. So it's not, one product is a platform, and you can, by changing a little bit the structure of the mRNA, the formulation, maybe the delivery route, we can make it always better.
So you would say that the benefits do outweigh the risk, in short?
Totally, yes. And I think, that is published, and again, if you have like one country or one place publishing clinical studies, okay, it's good, but you know that you have to look again. Now you have clinical studies have been done first in Israel, who has post-approval, in January 2021, people have continued in so-called phase 4 studies. So every country has its own way to do pharmacovigilance, to check side effects of even approved products. Medical doctors can report side effects they would see in their patients. And it's not centralized. Every country does it. So it's a safe vaccine.
mRNA wasn't just safe. It could be made at scale. Before COVID-19, production was tiny. We're talking about milligrams or grams. But during the pandemic, pharmaceutical companies ramped up to kilograms within months, even while battling supply chain chaos and logistical hurdles.
If you're interested in hearing about a successful scale-up of LNPs for mRNA vaccines, then we've linked to a webinar with process manufacturer Merck KGaA in the show notes.
So here's the big question. If mRNA can already work at scale and prove so effective, what else could it do?
Yes, and I think vaccines is the tip of the iceberg for mRNA. The part underwater is therapies, which is even bigger than vaccines because, as I said at the beginning, mRNA tells the cells what to do. So in theory, with mRNA, we can do anything. And I'm always saying there is a theoretical solution for any disease using mRNA. Any disease you can think about, autoimmune, autoinflammation, degenerative, injuries, even aging. Now, of course, from theory to approval, you need like many years and a lot of effort and money. But normally I ask people, challenge me, tell me a disease and I will tell you which mRNA in theory could help in Parkinson's disease, in Alzheimer's disease, in heart disease, in skin disease. And normally it's a game I like to do because then, you always end up to say, okay, with this mRNA coding for this protein, you could do that or that or that. And so in theory, the next years and decades will probably get approval of more and more therapies. We have seen already, a small baby, a newborn baby being corrected from a genetic defect using mRNA coding for enzyme that can repair DNA. Oh. And so genetic defects, everything can be, in theory, controlled by some mRNA or RNA formulations that you inject in people. And regenerative medicine is huge. So we have a program called RegRNA. It's the EU program to repair the heart after a heart infarct. And so we use mRNA that allow cardiomyocytes, so muscle cells of the heart, to proliferate, so to regenerate a muscle at the site of necrosis of the heart. So ultimately, this will work at one point, maybe not in 2 years, maybe not in 5 years, but let's say maybe in 10 years or earlier or later. It's always difficult to plan and to know when it will be successful. But at one point, it will be successful. So mRNA is used to regenerate the skin, the heart, the lungs, correct genetic disease, regenerate neurons. So the possibilities are nearly unlimited.
You're working with chemical synthesis of mRNA as well. What benefits does this offer over enzymatic synthesis?
This is mostly for cancer patients, because for cancer patients, what we are doing at the moment is to do individualized vaccine. So each cancer patient gets treated with his own product. It's totally individualized medicine. So what we do is that you take a biopsy of a cancer, and you make the genetic profile of this cancer. You find mutations compared to healthy cells in each individual. And then you make a vaccine that will allow your body to make immune response against the mutation of your own cancer. So this is in phase 3 studies by Moderna, also in studies by BioNTech. So each patient gets its own mRNA vaccine against his own mutations. So this is a process that takes like 3 months from the entry of a patient in the clinical study. You need to sequence the genome of a tumor. You need to sequence the healthy genome of the patient. You need to compare by informatic the two genomes, find the mutations, then conceive by informatic vaccine, and then produce it. And the in vitro production of mRNA, like it is done in the vaccines, in the approved vaccines, is easy to make it in big amount, but when you want to make it for each patient, it takes some time because you need people to be involved, to make the different steps of production. So you can make 1 gram or 1 kilogram with the same equipment and with the same people, but you need like 1 person per production, let's say. So if you have 20 patients, you may need 20 persons working on it, and especially some parts of the production needs to have people looking at it. By moving to chemical synthesis, we can have the production automated, because the chemical synthesis is fully automated on some kind of chemical synthesizers that are kind of robots. So I started to think about using chemical synthesis of mRNA in this context of individualized anti-cancer vaccines, because there's really a big workload, which means also a big price for making one vaccine for one person. So it's feasible, it's in phase 3, it's hopefully will be approved within 2 years, but it's big workload, it's big cost, it takes 3-4 months. And by moving from the normal enzymatic synthesis to chemical synthesis, we can reduce the time frame and go faster and sink the cost by having the whole production and formulation automated.
Remember how we talked about LNPs earlier? They were the breakthrough that made COVID-19 vaccines possible, helping mRNA reach lymph nodes and trigger strong antibody responses, something older formulations couldn't do.
The next challenge is designing LNPs that deliver mRNA precisely to specific tissues. Researchers are refining lipid chemistry and formulations, paving the way for more effective vaccines and therapeutic uses in the years ahead. Steve, I can imagine that the passion that you have for this topic has inspired at least some of our listeners. And if somebody is interested in being a part of the future of mRNA therapies, what advice would you have in terms of career and in terms of research focus? What is needed to advance this field, or who is needed to advance this field?
What I see is that collaborations are needed. So as I said, the chemists synthesize a lot of lipids, and then you need biologists and you need physicians, because a chemist will not know, for example, that the liver takes everything. A physician will know that the liver takes everything, but will not know how to get away from it. A biologist will know, lipid nanoparticle shape, structure, charge will dictate its biodistribution. So you need a few people to work together to manage to have solutions to this liver uptake, to the targeting. The mRNA itself will still improve it. And so it's kind of important collaborations, and to be horizontal. I think one person alone will not have the solution. You will need to work with colleagues from other fields to find new formulations, mostly to manage to treat the disease you want to treat.
So exciting opportunities for collaboration.
Yes, and also a lot of course, artificial intelligence to predict a little bit better what will happen to the RNA, what will happen to the particles. We still know very little what happens to mRNA. We think cells are full of proteins that interact with mRNA, full of RNA that interacts with RNA. We have long non-coding RNA, we have macroRNAs. We have a lot of things we don't know what they do, but they are there. Because we don't know what they do, we think they do nothing, but they do something. And when you make an mRNA going in the cell, it will interact with all these things in a good or bad way. We don't know. And only artificial intelligence can help us maybe better to predict, this mRNA sequence will interact with this protein and it will destabilize your mRNA. So you rather mutate here your mRNA in order to avoid to interact with this protein in this cell type. This is so complicated so far, we cannot predict it. But with artificial intelligence, kind of putting in place networks, from the cells interaction between RNA proteins, we can better say, now we will make this mRNA design and ask, is there a chance to the artificial intelligence, is there a chance that this mRNA will not perturbate the cell physiology, code for the protein for a long time, and by knowing better the macroRNAs, the long non-coding RNAs, the proteins, the structure of the mRNA, then we will manage also by prediction to make better mRNA. So artificial intelligence should be also part now of the decisions of how to make the mRNA, which sequence to make, and how to formulate it. So it's really multi-domain work that will need to take place.
We've been speaking with Dr. Steve Pascolo. He's a senior scientist in the Department of Dermatology at the University of Zurich. Kai, what were your key takeaways from this conversation?
Well, when we hear about mRNA, we usually think of COVID-19, but they have potential for treating so much more. Researchers have realized that you can treat a wide range of illnesses. There's some research done about regenerating the skin, the heart, the lungs, and even going into the area of genetic diseases.
But of course, if we think back that Steve was the first person to receive an mRNA injection all the way back in 2003, this research has been going on for a long time. And as an aside, Kai, I should say, I think Steve is probably going to be the only guest on this show whose skin has ever glowed.
Yeah.
But, for more than a decade after that, and of course leading up to that, there was all kinds of work being done on mRNA vaccines, and yet they were considered impractical because of stability issues. And then once COVID came around, we realized actually that's not the case, and it sort of entered our vocabulary at that point.
Yeah, people like Dr. Steve Pascolo really laid the groundwork, making sure that large-scale production is possible. This has been Balancing the Future from METTLER Toledo.
What questions about science and technology would you like answered in a future episode?
Let us know by leaving a review, or if you listen on Spotify, leave us a message in the comments section.
And be sure to subscribe wherever you get your podcasts.